As consumer preferences and legislation tighten around antibiotic use in agriculture, the poultry industry faces a mandate to find alternatives. Certain microbes maintain an asymptomatic presence in the birds, but threaten human food safety if the poultry products carry them. Reducing this pathogen reservoir is critical for pre-harvest safety. Selectively breeding for enhanced host immunity offers a permanent, cumulative solution to maintain flock health without relying on pharmaceutical intervention. This photograph shows an industrial poultry house, and it illustrates the massive population scale required for these interventions to actually take effect. But immunity requires massive metabolic resources. If you genetically program a bird to produce high levels of antibodies, you inevitably pull nutrients away from muscle deposition and overall growth. In the high hygiene environment of a research facility, this biological cost is a net negative for production. In a lower hygiene commercial farm facing active pathogen challenges, that robust immune response is essential to keep the birds alive. The core challenge in breeding for this balance is that an immune response is rarely dictated by a single genetic switch. These traits are highly polygenic, controlled by multiple loci scattered across the genome. Because immunity involves thousands of microvariables competing for limited metabolic resources, genetic selection is an architectural optimization problem. To map these variables, researchers look at the chicken genome's unique architecture, specifically microchromosome 16, which houses the Major Histocompatibility complex, or MHC. The avian MHC is highly compact and incredibly gene-dense. It is classified as the minimal essential MHC. This compact region features a high frequency of single nucleotide polymorphisms, or SNPs. This intense variation encodes the polymorphic proteins that give the bird a wide repertoire for antigen recognition. For decades, commercial breeders actively selected for specific MHC alleles to confer disease resistance. Yet, despite this targeted focus, the MHC explains only a small fraction of the total phenotypic variation we observe in a flock. To capture the missing variants, the field is abandoning the hunt for isolated markers and moving toward whole genome statistical prediction. To actively breed for complex immunity, we have to decode the statistical mechanics of how thousands of genomic tweets are inherited together over time. We begin with linkage equilibrium, or LE. Here, alleles assort entirely at random within a population. In linkage equilibrium, identifying a specific genetic marker provides zero predictive value regarding any neighboring functional gene. The chromosomes are thoroughly mixed. To successfully map quantitative traits, we require the opposite mathematical condition, linkage disequilibrium, or LD. In linkage disequilibrium, a specific marker allele and a functional trait allele exist together at frequencies much higher than random chance. Here, two parallel vector rods represent homologous chromosomes. A marked genetic locus sits in close physical proximity to a linked quantitative trait locus. During meiosis over successive generations, crossing over events occur, and the physical linkage between these two loci is randomly broken. The rate this physical linkage decays depends entirely on the genetic distance between the loci. Tightly linked loci retain their linkage disequilibrium for many generations. On the other hand, loosely linked loci lose their predictive correlation rapidly. Linkage disequilibrium is a decaying signal. Capturing it before it fades dictates our mapping strategy. The specific historical breeding background of a population dictates exactly the amount and extent of linkage disequilibrium available to measure. Historically, researchers mapped traits using F2 line crosses, mating two distinct breeds to generate large, predictable blocks of shared inheritance. On the left, the genome of an F2 cross is dominated by massive, unbroken colored blocks spanning wide regions of the chromosome. Because these unbroken blocks are so large, you can map them using a sparse grid of widely spaced data points. While sparse markers detect these massive blocks easily, they provide very poor positional resolution for locating the actual causative gene. Commercial breeding lines operate differently. These are closed, outbred populations with long, complex histories. On the right side, generations of continuous recombination have shattered those massive blocks into hundreds of tiny, highly fragmented slivers. The linkage disequilibrium has eroded. Predictive correlations now exist only between incredibly tight, adjacent loci. Capturing these highly fragmented signals requires up to 650,000 SNPs arrayed across the entire genome. This high-density approach is called a genome-wide association study. As the scale of commercial breeding grows, researchers abandon sparse positional mapping in favor of these hyperdense, population-wide association studies. For years, the industry relied on a two-step mathematical approach called marker-assisted selection. Marker-assisted selection's flaw is that it requires pre-screening SNPs for strict statistical significance before they can be used to calculate an animal's molecular score. This standard Manhattan plot maps an entire genome with hundreds of scattered data points. A few tall vertical peaks stand out as strong signals. In marker-assisted selection, researchers apply a strict threshold. Data points falling below this line are deleted, leaving only isolated peaks. Deleting this sub-threshold data eliminates the vast majority of the polygenic variants, specifically the thousands of genes that exert small, cumulative effects on immunity. The modern solution to this mathematical bottleneck is genomic best linear unbiased prediction, or GBLUP. By removing the arbitrary threshold, the deleted data points flood back into the model, and we evaluate the entire dataset simultaneously across the x-axis. GBLUP fits all SNPs across the genome simultaneously, without any significance pre-screening. The model treats all SNP effects as random variables, shrinking their estimates towards zero. This accurately models the true distributed genetic variance of the population. By mapping the entire genetic landscape, GBLUP transforms previously invisible polygenic variants into highly actionable breeding data for the future of poultry science. GBLUP solves the statistical challenge of polygenic selection, but it only observes mathematical association. It does not prove biological causation. Identifying the exact mechanistic pathways of host-pathogen interaction requires integrating genomic predictions with high-throughput multiomics. Through transcriptomics, researchers can map global changes in mRNA expression, specifically when a bird is subjected to an active pathogen challenge. The goal is to locate precise mechanistic targets, such as identifying a specific pathway that could be disrupted by an RNA decoy to block influenza viral replication. Statistical prediction tells us which birds possess resistance. Systems biology tells us how they achieve it, yielding the exact blueprints required for active engineering. In mammals, producing transgenic animals is structurally straightforward because DNA can be easily introduced into a single-cell oocyte. Poultry present a distinct physiological barrier. By the time an egg is accessible to researchers, the embryo inside already consists of over 50,000 cells. This side-by-side view illustrates the scale of the challenge, comparing a single dividing cell against a massive, rapidly expanding multicellular structure. To bypass this cellular barrier, researchers isolate primordial germ cells, or PGCs. These lineage-restricted stem cells can be extracted from the embryo and continuously propagated in vitro without losing their germ cell properties. In the lab, DNA transposons like piggyback or toll2 are used as highly efficient vectors, integrating the targeted disease-resistant sequence directly into these cultured PGCs. By combining whole genome statistical modeling with in vitro germline manipulation, researchers have bypassed the physiological limits of the avian embryo. The industry has shifted from observing the immune system to actively engineering it.